Hello everyone, I'm Theresa Comilang. I would be discussing the control of glucose levels, and afterwards I would also discuss the different disorders of carbohydrate metabolism. So, the blood sugar concentration, or also called as the glucose level, refers to the amount of glucose present in the blood of a human. Normally, in mammals, the blood glucose level is maintained at a reference range between about 3.6 and 5.8 millimeter or millimole per liter. It is tightly regulated as a part of metabolic homeostasis. So these are the basic considerations. The blood sugar or glucose concentration, the amount of glucose in milligram or in 1 deciliter of the human blood measured as milligram per deciliter or milligram percent. In the normal blood glucose, in the fasting state, it is 60 to 100 milligram percent, while in postprandial, it is 100 to 140 milligram percent. So these are the two disorder or a condition in the blood glucose level. The first one is the hyperglycemia. It is a condition in which an excessive amount of glucose circulates in the blood plasma. This is generally a blood sugar level higher than 11.1 millimole per liter or 200 milligrams deciliter. While the hypoglycemia, it is also a condition in which blood sugar or blood glucose concentrations fall below a level necessary to properly support the body's need for energy and stability throughout its cells. So I have an example here. I have these three circles. The first one is the hypoglycemia, which has the low sugar. The white one is the sugar. This is the normal level, which is has the normal sugar, and this is the hypoglycemia, which has a higher sugar. As you can see, there's a lot of small white circles, which is the sugars. So under the normal blood glucose level, in normal persons, blood glucose level is controlled within a narrow range. In the early morning, after overnight fasting, the blood glucose level is low, ranging between 70 and 110 milligram per deciliter of blood. And between first and second hour after meals, or also called as postprandial, the blood glucose level rises to 100 to 140 milligram per deciliter. Glucose level in blood is brought back to normal at the end of the second hour after the meals. Blood glucose regulating mechanism is operated through liver and muscle by the influence of the pancreatic hormones, which is the insulin and glucagon. Afterwards, I would be discussing the insulin and glucagon. So the next one, many other hormones are also involved in the regulation of blood glucose level. Among all the hormones, insulin is the only hormone that reduces the blood glucose level, and it is called the antidiabetogenic hormone. The hormones which increase blood glucose level are called diabetogenic hormones or anti-insulin hormones. So the necessity of regulation of blood glucose level. Regulation of blood glucose, or also called as sugar, level is very essential because glucose is the only nutrient that is utilized for energy by many tissues such as our brain tissues, retina, and germinal epithelium of the gonads. So the liver has a role for the blood glucose level, the regulation of the blood glucose level. Liver serves as an important glucose buffer system. When blood glucose level increases after a meal, the excess glucose is converted into glycogen and stored in the liver. Afterwards, the blood glucose level falls, the glycogen in liver is converted into glucose and released into the blood. The storage of glycogen and release of glucose from liver are mainly regulated by insulin and glucagon. So the hormonal regulation. There are two types of mutually antagonistic metabolic hormones affecting blood glucose levels. The first one is catabolic and the second one is anabolic hormone. So catabolic, such as glucagon, growth hormone, cortisol, and catecholamines, which increase blood glucose, while anabolic hormone, such as insulin, which decreases blood glucose. So the role of pancreas. The pancreas detects the change in blood glucose concentration and releases the appropriate hormones. So in here, as you can see, if you have a high blood glucose, it goes to the pancreas, down to insulin released by beta cells of pancreas, down to the fat cells take in glucose from blood, and now it achieves normal blood glucose levels. And if you have a low blood glucose, it goes to the pancreas, down to the glucagon released by alpha cells of pancreas, down to the liver releases glucose into blood and now achieve the normal blood glucose level. The roles of insulin and glucagon in regulating blood glucose. Insulin and glucagon are hormones that help regulate the levels of blood glucose or sugar in our body. Glucagon binding to its receptors on the surface of liver cells triggers an increase in cyclic adenosine monophosphate production, leading to an increased rate of glycogenolysis by activating glycogen phosphorylase via the pKa-mediated cascade. This is the same response hepatocytes have to epinephrine release. The resultant increased levels of G6P in hepatocytes is hydrolyzed to free glucose by glucose-6-phosphatase, which then diffuses to the blood. The glucose enters extrahepatic cells, where it is rephosphorylated by hexokinase. Since muscles and brain cells lack glucose-6-phosphatase, the glucose-6-phosphate product of hexokinase is retained and oxidized by these tissues. Under the insulin, insulin stimulates extrahepatic uptake of glucose from the blood and inhibits glycogenolysis in extrahepatic cells and conversely stimulates glycogen synthesis. As the glucose enters hepatocytes, it binds to and inhibits glycogen phosphorylase activity. The binding of free glucose stimulates the dephosphorylation of phosphorylase, thereby inactivating it.